Welcome again to iTTV's lesson on Biology Form 4. We are still in chapter number 2, and today we'll be looking at cell organization. I'm Mr. Gary to take you through our entire adventure of biology. Now, living organisms can either be unicellular or multicellular, right? You've learned this back when you are in PMR. Now, what is the definition for a unicellular organism or a unicellular microorganism? Now, a unicellular microorganism consists only of one cell. However, a multicellular microorganism or an organism can consist of more than one cell. So take for example, your pet, a cat or a dog or a rabbit for example, or even your hamster for example. Is it a unicellular or multicellular organism? Yes. These organisms are multicellular because they have more than one cell, and therefore we have specialization in cells occurring to them. However, in today's lesson, what we are going to see is we are going to check out on several unicellular organisms which are very, very important for you. Now, back when you were in PMR, the unicellular microorganisms that you saw were paramecium and amoeba. Now, I would like to take your attention to these two freshwater living creatures. You see, we can't see them through our naked eye. Therefore, we can only observe them through microscopes. Therefore, these unicellular microorganisms might elude our vision. However, they do exist. And because they are also living things, therefore, they can carry out all living processes like how we do. So what are these things that we call living processes? They can feed, they can respire, they can excrete, they can move around, they are mobile, okay? They can even grow, and they can also reproduce. Therefore, if there is any cells or any organism which can carry the following of the living processes, then we consider them as a living organism. Take for example, this pencil. Do you think the pencil can move? Can it respire? Can it excrete? Can it reproduce? It can't do any of this. Therefore, we consider this pencil as a non-living thing. So now you know the difference between a living and a non-living. Now let's take a look at the living lifestyle of two unicellular microorganisms that were so popular, amoeba and paramecium species. Now both of them are protozoans. Okay, let's take a look. First we will start off by looking at the significance of the amoeba species. Now take a look at the name. We have written it as capital A, M-O-E-B-A, and then space, we have S-P dot. This is the way you will have to write down the names. Now, this form of name was given to us by a man called Linnaeus. So we call this as a binomial Linnaeus method, which you will learn towards the end of our Form 4. Now, according to Linnaeus, he said that every name of organism can be classified into the genus and the species name. So for example, in our amoeba SP, the name of the genus is amoeba, but we are not sure of the name of the species. So what do? We just write down the initials SP dot. Now remember that you'll have to space them out, and remember that you'll even have to underline each one of those words. So students, let's see what is the right way we will have to write the names. Now, take for example, I would want to write down the name of amoeba SP. So this is the way I will have to write down. A capital A, M-O-E-B-A. Now this is the name of the genus. And because I'm unsure of the name of the species, I will just have to write the letters or the initials SP and have a dot. Now you see, we can't just write them and leave them like this. We will have to even underline them. So remember that you will have to underline them with a space in between, not a continuous line. Now, if you did not do it the right way, say for example, you wrote it down as amoeba SP dot and I underlined them. Now, this is a wrong statement, the wrong method of writing down. Why? First, there is no capital A for the genus, and here we can see that the underline is continuous. So pay attention from now onwards. Every time you want to refer to a genus or a species, you will have to write it this way, not this. Okay? Let's carry on with our amoeba. So students, Amoeba is an aquatic organism that could be found in freshwater environment such as ponds, lakes, or in soil water. It has an irregular shape, therefore it has no definite shape. Why doesn't it have a definite shape? Because of the absence of a cell wall. Why? Because this is an animal cell, not a plant cell, right? Good. The organelles, like food and contractile vacuoles, are found in the cytoplasm and are enclosed by a plasma membrane. The cytoplasm is further divided into two parts, the outer layer called ectoplasm and the inner layer called the endoplasm. The amoeba is a living organism, therefore it's got to have a nucleus which controls its cell activity. So how are these different types of cytoplasms placed in them? How can there be two types of cytoplasm? Let's take a look at the diagram, okay? Now this is the basic structure of an amoeba species. Now as you can pay attention on the top, you can see that there is a cytoplasm and there is an endoplasm and ectoplasm. So on each of the projections that come out from the amoeba, that projections contain the ectoplasm. Ecto referring to outside and endo referring to the inside. Okay? Let's carry on. You can see that there is a food vacuole there. A food vacuole stores food, obviously. And then we can find a contractile vacuole. Now, what is the function of this contractile vacuole? You will see this again in the paramecium species. A contractile vacuole helps the amoeba or the paramecium later on to regulate its own osmotic pressure. Because if you pay attention, we will know that the amoeba and paramecium are found in fresh water. Therefore, water can move into the cell and out of the cell because the plasma membrane cannot control the movement of water molecules itself. Therefore, what will happen if there is an influx, or let's say there is too much of water coming in from the environment into the cell? The cell will eventually bloat and it will swell up and it will eventually burst. And your amoeba will cease to exist. So one way the amoeba excretes this water is through the contractile vacuole. So the contractile vacuole will absorb the excess water that moves into the cell. And as the size of the contractile vacuole increases, once it reaches its maximum size, it will contract. Now do you know why the name is contractile vacuole? Because it can contract and it will expel the excess water out. Alright, let's carry on. Now you can also see that there is a presence of a nucleus and a plasma membrane, which is not so important at this moment, but pay attention to something known as the pseudopodia. You can observe the pseudopodia, right? They are projections that look like arms, which act as arms for the amoeba. So the word pseudopodia comes from a Latin term and is divided into two derivatives, pseudo and podia. Now the word pseudo, the meaning of that word is actually false, in other words, bluff. Okay, that means it's not true. The word podia, however, refers to limbs. Limbs, for example, hands and legs. Now if you can see, the amoeba can move. The amoeba can also feed. That means it can eat and it can move. However, it doesn't have feet or legs and it doesn't have a hand. For example, if we wanted to eat, we will use our hands to gather the food and eventually use a structure called the mouth and bite it into our body. However, the amoeba does not have that, right? So how does it gather food? It uses a false limb by projecting its cytoplasm. So imagine a normal animal cell, and if it can project its cytoplasm, it would form a limb, and that's how it would gather the food. Once again, let's go back to the diagram. You can see now for sure how it works. Check it out. So you can see that there is an empty space between the pseudopodium, right? Now food will move into that empty space and eventually the pseudopodia will close up into it and it will gather the food into its body. We will see detailed diagrams in a while. Let's carry on. The amoeba carries out respiration as well. Now, respiration in the amoeba involves the exchange of respiratory gases such as oxygen and carbon dioxide by simple diffusion through the plasma membrane. Now, if you take a look, there is a plasma membrane and respiratory gases simply diffuse directly into the amoeba through the plasma membrane. Next, the amoeba can also move about. Therefore, it can carry out locomotion. The words locomotion mean mobility or movement. The amoeba moves by extending its pseudopodia or the false feet by anchoring the tips onto the ground. The cytoplasm then flows into these extended pseudopodia, while the others not in use are withdrawn. This type of movement is called the amoeboid movement. So remember, the amoeba moves in what fashion? The amoeboid movement. Now basically, long story short, okay, what is this amoeboid movement? It will simply project its cytoplasm forward, and it will use this pseudopodia or false feet to point its tips to the ground and pull itself closer. Get it? Let's continue. As you can see, I've put for you a step-by-step guide on how it moves. Now check out the first diagram. You can see that it moves by extending the pseudopodia in the direction of its movement. And as the cytoplasm flows into that area, a pseudopodia is formed. Next, the cytoplasm flows into this extended pseudopodia, whilst the others not in use are withdrawn. Therefore, you can see that the cytoplasm is flowing from the left to the right of the amoeba. And eventually, the amoeba has made a certain type of movement. This movement is known as the amoeboid movement. Let's now take a look on how the amoeba feeds. Okay? The amoeba also uses its pseudopodia once again for feeding. It feeds on algae, bacteria, and other microorganisms. Remember, the amoeba feeds on bacteria and microorganisms because it is an animal cell. The amoeba feeds through a process called phagocytosis. Now students, this is very important for you to know. Phagocytosis is not only carried out by amoeba, but few other organisms or microorganisms and even white blood cells in our body. So pay attention to phagocytosis, as you will keep seeing them throughout your Form 4 and Form 5 biology lessons. Now, the presence of food causes the amoeba to move towards the food. It extends the pseudopodia to engulf the food to form a certain food vacuole. Once the food is ingested, the hydrolytic enzymes called lysozymes in the cytoplasm is secreted from the lysosome to digest the food. The nutrients from the food are then absorbed, whilst the undigested food is expelled away from the body. Now let's take a clearer look using a diagram on how the feeding mechanism of an amoeba is. Now remember, what process does it use? Phagocytosis. Good. Now check out the food molecule which is found in between two pseudopodia. Now the pseudopodium eventually will gather around the food molecule, and we call this as engulfing the food molecule. Once it is engulfed, it will join. Which will join? The pseudopodia will join together and form a continuous cytoplasmic region. Therefore, now the food molecule will eventually be trapped inside the amoeba. How wonderful, doesn't it sound? The amoeba simply engulfs the food and it gathers its cytoplasm once again, and now the food molecule is trapped inside. The moment the food molecule is trapped inside, what does it do? Still remember the structure called lysosomes? Yes, they have hydrolytic enzymes, right? Called what? Lysozymes. Now that lysozymes will be secreted onto the food and they will digest the food. Whatever nutrients it needed, it will absorb directly into its cytoplasm. Next, see the step-by-step guide of phagocytosis. You can see the amoeba is moving towards the food molecule because the food has already been detected. Next, the pseudopodia tries to engulf the food. And once engulfing, you can see on the third diagram that it is closing in completely. And on the fourth diagram, what has happened? It has already completely carried the food molecule into its body. And while it's doing that, it has already detected another food molecule, right? Good. So this is how an amoeba feeds. Next, if it has to feed, then it also has to excrete. Same like human beings as well. Now let's see the excretion found in amoeba. Waste products such as carbon dioxide through respiration and ammonia are excreted by simple diffusion through the plasma membrane. The contractile vacuoles in the amoeba is involved in osmoregulation. Now what is osmoregulation? Osmoregulation is the control of the osmotic pressure of the cytoplasm of the amoeba species. Next, water diffuses into the cell and fills the contractile vacuole. When the contractile vacuole is filled to its maximum, the contractile vacuole then contracts to expel the contents, and this is done regularly in the life cycle of an amoeba. So let's take a look at the diagrams which involve the excretion of amoeba. As you can see in the first diagram, you can see the presence of a contractile vacuole found in the cytoplasm of the amoeba. Waste products are excreted by diffusion through the plasma membrane. The contractile vacuole is also involved in a process called osmoregulation, whereby you can see the arrows are showing you that water molecules are moving into the cell. As water molecules move into the cell, the maximum size of the vacuole has already increased. Now, water diffuses into the contractile vacuole, and when it's in its maximum stage, the vacuole will then contract and therefore discharge whatever content it has, which in this case is the water molecules, into the exterior environment. Now, the vacuole returns to its normal size, and it has already helped the amoeba in osmoregulation. Let's carry on. The amoeba is a living organism. Therefore, it can respond to any form of stimuli. Now, what is stimuli? Stimuli is an external factor that causes the amoeba or any other species in that example to move or to respond towards it. For example, take yourself. If your mom would come and give you a hit or a slap on your arm, would it hurt? And if it hurts, after your mom hitting or tapping your shoulder, do you just keep quiet and do not make any movement? You don't, right? A normal response towards this form of stimuli would be ouch or stop it. Don't do that. You are responding towards it. True? Let's say, for example, you're fast asleep, and the moment you wake up, your brother or sister opens the window shades and allow the sunlight to penetrate into your room. What would you do? You would also respond to it. How? By crouching at it. Oh, it's very bright. I can't take a look. These are responses that you give towards certain stimuli, for example, pressure, light, heat, pain. All right? Now, if you can do it, so can unicellular organisms, because they are alive too. Let's take a look at how the amoeba responds. The amoeba responds to stimuli by constantly changing its shape. Wonderful, isn't it? When an amoeba senses a favorable stimulus, for instance food, it moves towards it. However, if the stimuli is unfavorable, such as water that is acidic or alkaline in nature, bright light, and high temperature, it moves away from it. So you can see how the amoeba responds. Once again, let's recap. What does the amoeba respond to? Light intensity, food, pH of environment, and even temperature. So if you've got these four in you, that's more than good enough. Okay? Now let's take a look on how the amoeba would reproduce, because it's a key aspect of living organisms. If they can't reproduce, they can't populate, and therefore they will cease to exist, right? Let's take a look. The amoeba reproduces asexually. When the conditions is favorable, it produces by binary fission. However, when the condition is unfavorable, it reproduces by spore formation. Now, what is this thing called binary fission and spore formation? Now just pay attention to the words fission. Now there are two words that sound similar: fusion and fission. Now fusion means when you take two molecules or two parts and join it together, you're fusing them together. However, if you break them up into two, then you call them as fission. Now the amoeba can carry out binary fission. Binary means two. So one amoeba can split into two amoeba. Wouldn't it be great if we could do that? Next, let's take a look at how binary fission happens. You can see that the parent cell begins to divide in the first diagram. Pay attention to the nuclei, because the nucleus is the one that would begin to divide first. Now on the third diagram, we already have two nucleus. Therefore, cell division is almost complete. However, because the cytoplasms are joined together, they are still not considered as two individual microorganisms. And now pay attention to the fourth diagram. You can see that the cytoplasm is beginning to cleave or beginning to divide. However, in the fourth diagram, you can see that the division of the cell is almost complete, right? Now, the moment the plasma membrane is divided into two and the cytoplasm is separated, we will end up having two individual amoeba. However, this form of reproduction is known as asexual reproduction. Therefore, there is no variation. Therefore, both the individual daughter cells are identical to the parent. Now, we will see more of this in another chapter called cell division. It's coming up. Just be patient, okay? Next, let's take a look at a more beautiful diagram of the binary fission occurring in an amoeba. You can see in this color diagram how the nucleus start dividing, and then they form two separate nuclei, and then you can see the cytoplasm and the plasma membrane cleaving, and eventually the end of this would be in two individual amoeba. So we have already seen how the amoeba carries out all its living processes, right? Now, it's important for you to remember the basic ideas of the amoeba and how it carries out its living processes. Now, this animal cell is unique. Now, let's see if we really remember everything. Okay, let's go to our assessment on amoeba. Question number 1. A student observed that the amoeba species doubled its number every 30 minutes. Name the type of reproduction observed. And next, draw the process of reproduction as you have named in one above. So first, the type of reproduction observed. Is binary fission. You are right, because it doubled up, right? And this is how the drawing would look like. A single cell divides its nucleus into two, then the cytoplasm starts to cleave and the plasma membrane starts to cleave. It's almost complete, and eventually it separates into two individuals. Next, the false feet of the amoeba species is used to trap food and to move around. What is the name of the false feet, and explain its meaning? The false feet is called pseudopodia, and they are cytoplasmic projections. Question 3. How does amoeba ingest their food? The amoeba feeds through a process called phagocytosis, which is extending its pseudopodia to engulf the food and form a food vacuole. Now students, the word ingest doesn't mean digest. Ingest means taking the food from the outside into its body. How do humans ingest? By using our mouth. Take for example a burger, you bite into the burger and you take it into our body. That is ingest. So we are only talking about ingestion in the amoeba. So I believe you have got all the ideas, and I believe that you could carry on with an assessment. And I hope you really guessed before I gave you the answers. Now, if you could do that, you are on track and you have a very good interest in biology. Keep it up. You might become somebody very important in the biology of the world itself. So students, that's all for amoeba species. Now, in the next lesson, we'll be looking at paramecium and how it carries out its living processes. So until then, thank you for watching iTTV.